Have you ever wondered, what is even, have we ever wondered why an elephant always gives birth only to a baby elephant, right? And not same other animals? Or why a mango seed forms only mango plant, right? And do not any other plant? Why? So this time we know, because their genes are, mango genes is there, elephant genes is there. If that they do, are the offsprings identical to their parents? Okay, sometimes we said them they are identical to their parents because they have same organs are there, right? Mango means it is a only single seed is there, right? And fruit is also drupe fruit is there. In all mangoes, drupe fruit is there. They have a thick stem. Same tissues are present, but they have some variation is also there. But they have variation is there because no two one we can see in human also. No two individuals are same or identical except monozygotics. That is given here. Or do they show differences in some of their characteristics? Yes, some characteristics they have some differences also. Have you ever wondered why siblings sometimes look so similar to each other? Why siblings they look similar to each other, or sometimes even so different? It is depend upon the genes, right? These are several related questions are dealt with scientifically in a branch of biology, and this branch of biology is called as genetics. This subject deal with the inheritance, that is heredity, as well as the variation of characters, that is variation from parents to offspring. Already we know. Heritance is the process by which characters are passed to the next generation, right, from parent to progeny, and it is the basis of heredity. That is called heredity, right? What is heredity? Process by which the characters are passed from parent to offspring. Second, variation is the degree by which progeny differ from their parent. Variation, it is the degree by which progeny differ from their parents. That is variation. So, what is genetics? Genetics is the, we can say, collectively study of variation and inheritance, or heredity and variation. Human knew from as early as 8000 to 1000 BC, before the birth of Christ, that one of the causes of variation was hidden in sexual reproduction. At that time they understand, right? They exploited the variation that were naturally present in the wild population of the plant, because at that time they start to grow some plants, right? So they collect, that is, we can say, domestic domestic varieties from the wild varieties. At that time they collect, they separate, and they also knew about the that is wild. Here they exploit the variation that were naturally present in the wild population of plants and animal to selectively breed, right, and select the for organisms that possess desirable characters. At that time they knew breeding, right? Okay. For example, through artificial selection and domestication from ancestral wild cows, we have well-known Indian cow, that is Sahiwal, Indian breed, that is Sahiwal, cows in Punjab, that is the best cow in our country. We must, however, recognize, best cow means Sahiwal, best buffalo means it is Murrah. We must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of genetic inheritance. That is this phenomenon. They have no idea about the scientific reason, but they knew about the breeding. Mendel's law of inheritance. Second point is Mendel's law of inheritance, right? Here some figure is given and you have to read this figure. 7 pairs of contrasting traits in pea plant studied by Mendel. Mendel read these 7 pairs of traits, right? These are dominant trait, recessive trait. That means they are alternatives. Seed shape, round and wrinkled, right? So seed shape, when we consider the seed shape, which this seed shape, round and wrinkled, and this gene is present in 7th chromosome, right? 7th chromosome. Now seed color, second is our seed color. When we see the seed color, the seed color is present on first chromosome. First chromosome, this yellow and green, dominant trait round and wrinkled, yellow and green. Flower color, violet or white is given here, right? When we see the flower color is means this one is also present in color, both color they are present in first chromosome. Violet dominant, white recessive, right? Now second one is our pod shape. Pod shape, it is the shape of pod, not the color of pod. The shape of pod means it is present on fourth chromosome. Then full and constricted, the full means what? It is a dominant, right? Then pod color, the pod color of pod, it is located on fifth chromosome. Fifth chromosome, okay. Look, seed shape 7, SS7, seed color and flower color on first chromosome. Pod shape, it is on fourth chromosome, and pod color means it is yellow and green, right? When we see the pod color, that green is dominant, right, and yellow is recessive, okay. Now here, flower position and stem height. When we see the flower color and stem height, this flower position, it is located in fourth chromosome, right? These both fourth chromosome, and this stem height, it is also located in fourth chromosome, right? Pod color means, pod color it is given here, fifth chromosome, right? Here you understand three, that is first, then fifth, then 7, right? First 4, we can say here, how many chromosomes are involved in these 7 traits? It is first, then 4, then 5 and 7 chromosomes are involved in these 7 traits. 7 traits, 1, 4, 5, 7. Do you understand here? Flower position is axial is the dominant and this one is the terminal. Okay, now here Mendel's law of inheritance. It was during the mid 19th century that the headway was made in understanding of inheritance. Mid 19th century, right? Mid 19th means it is start from 1856. So 1856 we can say it is the mid 19th century. Gregor Mendel conducted hybridization experiments on garden pea. Garden pea means was Pisum sativum. Scientific name is Pisum sativum, right? Garden peas for 7 years from 1856 to 1863 and proposed the law of inheritance of living organisms. During Mendel's investigation into inheritance pattern, it was the first time that statistical analysis and mathematical logic were applied to problem in biology, and that is the reason of, means we can say, for failure of Mendel. Mendel is not failed, but he is not means he did not convince the scientist and his contemporary scientists, right? They are not agree with this one. His experiments had a large sampling size which gave greater credibility to the data that he collected. Also, the confirmation of his inference from experiments on successive generation of his test plant. Test plant means what? It is Pisum sativum. Proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the Pisum sativum plant that were manifested as two opposite traits, or we can say complementary traits, or we can say they are alternative traits, right? That, for example, tall or dwarf plant, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observation and the complexity inherent in them. Okay, you have to read this passage, right? Because statement type question come, then they come from this one. Mendel conducted such artificial pollination. Mendel conducted artificial pollination. Artificial pollination, that is cross pollination experiments using several true breeding pea lines. True breeding pea lines. True breeding means it is homozygous, capital T, small t. True breeding pea lines means what? When we sow these plants, right? What happens in the offspring? Same type of plant come. Many is given here. A true breeding line is one that having undergone continuous self-pollination shows the stable trait inheritance and expression for several generations. Stable traits. Mendel selected 14 true breeding pea plant varieties. 14 because 7 contrasting traits are there, right? How many pure breed lines are there? It is 14, right? Tall, dwarf, right? For one trait, 2 plants, 2 true breed lines. As pairs which were similar except for one character with the contrasting traits, right? Some of the contrasting traits selected were smooth or wrinkled seeds, as for example, smooth or wrinkled seed, yellow or green seeds, right? Inflated full or constricted green or yellow pods, these two. Constricted or this inflated full pods, green or yellow pods, tall and dwarf plants. This is given here. We already understand, right? These are the traits, contrasting trait which is selected by the Mendel. 7 traits. Contrasting traits studied by Mendel in pea. What are the traits is given here? You can say stem height, tall and dwarf, flower color, violet and white, flower position, axial terminal. Here, pod color means pod shape means inflated or constricted. Pod color green or yellow, seed shape round and wrinkled, seed color it is yellow or green. This side it is given tall, dominant, violet, dominant, axial, dominant, inflated, dominant, green, dominant, round, dominant and yellow, dominant. And this side they are recessive traits. And this side is dominant trait. Right? These are the contrasting traits studied by the Mendel. Now it is second point is inheritance of one gene. Inheritance of one gene means what? It is monohybrid cross. This one is termed as monohybrid cross. Right? Here you understand how he performed his experiments, because we know pea means that is our Pisum sativum. It is a bisexual plant, right? Bisexual plant. It contains both androecium and gynoecium, right? So first he used emasculation. Emasculation means what? He removed the anther before maturation. He removed the anthers. Then bagging. Emasculation and bagging he used here, right? So first he removed the anthers, then bagged. Bagged means he covered the gynoecium with a polythene bag, right? So that new or foreign pollen grains are not reach at the pistil, right? We can say at the stigma. Then artificially, right? Then the desirable pollen grains he is brushed on the stigma. That is given here. Petal, stigma, anther, stamen, carpel. You understand this one is because it has androecium and gynoecium both are there, and we know this Pisum sativum, it belongs to a leguminous plant, right? Fabaceae family. So first, what happens? These anthers, they should be removed. The second point is given how he used removal of anther. That is called emasculation, right? So first, with the use of this sap, look, he cut the anthers. Anthers are cut down before maturation, right? Then what happens, right? Here, anthers, transfer of pollen and pollination. Now, from a new plant, that is white plant. You can say here, it is a white flower, that is the violet flower, right? So he transfer of pollen grain from white flower to the stigma of the violet flower, right? Then what happens? He get a new variety, that is the pea, right? And when he show these seeds, then he get all the plants with violet color. Then he understand this trait, white color trait, is not transferred. He simply thinks it is not transferred in the next generation. In F1 generation, he understands, right? But in F2 generation, then he understands that these are present, this white color trait, it is present inside the plant, but unexpressed. Okay, here the first monohybrid cross by Mendel, right? So monohybrid cross by Mendel means what? Single trait. Monohybrid means single traits. By using emasculation and bagging, emasculation and bagging method, right? Because P, that is Pisum sativum, is a bisexual plant. Emasculation means what? Removal of what? Androecium, stamen or androecium before mature, right? Emasculation by scissor, then bagging. After emasculation, this stigma, or we can say pistil or stem, is covered by bag. Polythene bag we use also, polythene bag, so that unwanted pollen grains never reach to stigma. That is called bagging, right? So what he do with this one? He used capital T, capital T, capital T crossed with small t, small t. That is the tall plant, and this one is dwarf plant, right? And these are parents, right? When he crossed these two, then he in the first capital T, small t, F1 generation, right? This F1 generation is formed. F for filial first generation. F1 means filial first generation. Filial first generation. That is the full form. F1 generation. F1 generation means all the plants are tall, phenotypically tall. What is the phenotype of F1 generation? We can say phenotype tall, right? Now, when in F1 generation all the plants are tall, then he first, what is the conclusion of this first generation? He gives, that means it proves this generation, it proves law of dominance. Law of dominance. How it proved law of dominance? This law of dominance. How it proved? Because in F1 generation only one trait is observed. Only one trait is appeared. Another trait is not appeared. Which trait is appeared? That tallness. Tall is appeared, but dwarf is not appeared. Genotype means both 50-50% genotypes are there. 50% genotype is capital T, 50% genotype is small t. But in F1 generation only tall plant is there. That means what? Tall is dominant over dwarf, right? So this principle is called law of dominance. It is the first law of Mendel, right? First law of genetic inheritance. Mendel's first law is law of dominance. Here, what is according to law of dominance? When a cross made between two different traits, two complementary traits, two contrasting traits, as for example, capital T and small t means between tall and dwarf. In F1 generation, only one trait will be expressed and another cannot be expressed. The trait which one is expressed, right? This one is called dominant trait, and trait which is not expressed, that one is called recessive trait, and that law is called law of dominance. When he right now, when he selfing is what? No other hybridization is there, right? Through selfing, when he selfing this one, then what happens? He found four seeds. Which one? Here capital T, small t, capital T, and small t, small t. Types of plants. Phenotypically means this one is tall, this one is tall, this one is tall, but this one is dwarf. This one is dwarf, and these three, they are tall, right? Tall, because they are tall. So what is the ratio of phenotype? Phenotype ratio is here 3:1. These three, 1, 2, 3:1. You can say 3:1. That is the phenotype ratio. But when we consider genotype ratio, then we get here, you understand, 1 tall 3:1. Here 1 tall Okay, I change this color, otherwise you can get confused. One tall, then two they are hybrid, and one, this one is dwarf. So 1 is to 2 is to 1, this one is 1 is to 2 is to 1, this one is phenotype ratio, and 3 is to 1 is the genotype ratio. 1 is pure tall, 2 are intermediates, means hybrid, and 1 is pure dwarf. Okay, if he is selfing, means this is called F2 filial second generation, right? So in this filial second generation, what he concluded, that in filial second generation, in F2 generation, they are segregate. So here this represents law of segregation, or we can say law of purity of gametes. These two laws. Here he said in SF means F2 generation, what happens? They segregate. Segregate means what? They separate. Means both are present in this F1 generation, both are present in the same, right? But in different, what happens? These gametes in the formation, we can say in the formation of gamete, what happens? These genes, they get separated, right? That one is called law of segregation. And here in F1 generation, what happens? They are present in the same, right? But they did not disturb anyone others. They are not intermingled, we can say here. So these gametes are segregate in their pure form. So this is also called as law of purity of gametes, and it is the second law of inheritance. Second law of inheritance. This one, law of dominance, it is the first law of inheritance. This one, law of dominance, and these two, they are second law. So through monohybrid cross, here law of dominance and law of segregation were proved, right? Okay, how he understand that this one is pure and this one is pure, right? So when he selfing these seeds, means in all the generation, these seeds it give only capital T, capital T, capital T, capital T, so on. This one is also only give small t, so on, right? No variation is there. But when he use this or this one, right, so he get once more the same capital T, capital T, capital T, small t, capital T, small t, the same ratio which he get 1:2:1 genotype, or 3:1 phenotype, right, in another generation. Do you understand here? Means here also law of segregation happens. This one is called F3 generation. Okay, do you understand? Monohybrid cross. You can see here. Monohybrid cross, right? By using emasculation and bagging, right? He made this experiment between tall and dwarf plant, right? F1 generation means it is all the plants in F1 generation, all the plants are tall, but in F2 generation they segregate with each other, right? And F2 generation means we have 3:1 ratio of phenotype, and genotype means it is 1:2:1 genotype is there, and through this ratio we get understand that law of segregation means here what happens? Here these seeds are segregated, these seeds not, these genes are segregated, these factors are segregated independently, or we can say purity. It is purity, law of segregation law. Independently is not given here. Independently is proved by dihybrid cross, right, which we are going to read. One thing I want to tell you, which is important, right? Then after we will go to dihybrid cross. What is it? How could we understand how many genotypes are there, how many phenotypes are there, right? Or we can say how many gametes are produced, number of zygotes are produced, right? So we have some law is there. It is not a theoretical law, but it is only a trick, right? How we get this. So here, so how many genotypes are there? For phenotype, TYPE, number of phenotypes. We should write here number of phenotypes. The number of phenotypes means we use n square. Sorry, 2n, right? Second, it is a number of genotype. It is 3n simply. N for traits. And when we consider number of zygotes, right, this one is 4n. N indicates here n for number of traits we use, right? As for example, suppose I am asking in a trihybrid cross, number of genotype and phenotype. What is the number of genotype and phenotype? Can you tell me easily by using this formula? You can easily tell me. So genotype means what? Genotype, I write here genotype means 3n, and we use trihybrid cross. Trihybrid cross means what? 3n for 3, right? Now we get 3 multiplied by 3 multiplied by 3. Now we have 27 genotype. 27 genotype in trihybrid cross, right? And when we consider the phenotype, phenotype means 2n. We use 2n. 2, 3 because trihybrid cross is there. It is 3. Now we get here what? We get 3 multiplied by, sorry, it is 2, right? 2 multiplied by 2 multiplied by 2. Now we get 2 to the 4 to the 8 phenotypes. Simply, how many zygotes are formed? Zygotes. So we understand here because we use 4n. So 4n means what? 4, 3. We should write here 4 multiplied by 4 multiplied by 4. Then what we get? 4 4 is 64. 64 zygotes are formed. So simply you understand how we calculate the number of genotype or phenotype by these simple tricks. This simple trick. Very easily. Suppose that even 5, it is a that is trihybrid cross, pentahybrid cross. Pentahybrid cross. How could you understand how many is types of, how many phenotypes are there? So penta means what? So then you use this one. Penta means, phenotype means what? 2n. N means, penta means 5 for pentahybrid cross. Right? N means what? 5. Means 2 multiplied by 2 multiplied by 2 multiplied by 2 multiplied by 2. Then we get 2 to the 4 to the 8 to the 16 to the 32 phenotypes are. We get 32 phenotypes. Simply, when you use this law, you easily calculate the number of phenotype or number of zygote or number of genotype we get understand. Okay. Now come to the NCERT. here. Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by the Mendel, where he crossed tall and dwarf pea plant to study the inheritance of one gene. He collected the seed produced as a result of this cross, collected these seeds and grew them to generate plants of the first hybrid generation. Right? This first hybrid generation. What he find this generation is also called as filial first progeny or F1 generation. Mendel observed that all the F1 progeny are tall, like one of his parents, right? None were dwarf. No one is dwarf, right? He made similar observation for the other pairs of traits also, because he used these 7 traits, and these 7 traits means one is dominant, another one is recessive, right? He found that the F1 always resembles either one of his parent, and that the trait of other parent was not seen in the F1 generation. That's why he gives first law of inheritance, that is law of dominance, right? Means one, the trait of, here given, he found that F1 generation always resembles either one of the parent, that parent is called dominant traits it contains, and the trait of the other parent was not seen, this one is called recessive, and this law is called law of dominance. First law of dominance, law of inheritance, that is law of dominance. Okay. Mendel then self-pollinated the tall F1 plant, and to his surprise, self-pollinated means through selfing, it is called, that in the filial second generation, some of the offsprings were dwarf. Some is 25% are dwarf, 75% they are tall. So this is called phenotype ratio 3:1. The character that was not seen in F1 generation, it is come into the F2 generation. That means what? They are separated, they are segregated, right? Then second law is coming, law of segregation. The proportion of plants that were dwarf were 1/4 for the F2 plants, while 3/4 of the F2 plants were tall, right? The tall and the dwarf trait were identical to their parental type and did not show any blending. Capital T, small t, right? Only 2 means which is hybrid, there are some blending there. That is, all the offsprings were either tall or dwarf, none were of in between height. 3 means total tall, equal length of tallness, and 1 is small, dwarf. Similar results were obtained with the other traits that he studied. Only one of the parental trait was expressed in F1 generation, while at the F2 stage, both the traits were expressed in the proportion of 3:1, phenotypically proportion, right? And the contrasting traits did not show any blending. They did not show any blending. That means what? That is the second law, which is called law of segregation. They get separate, that is law of segregation. And because no blending is there, so this one is called as law of purity of gametes. No blending, pure gametes. Law of purity of gametes. Right? Based on these observations, Mendel proposed that something was being stably passed down from one generation to another generation unchanged, means in pure form, from parent to offspring to the gametes over successive generation. He called these things as factor, right? Now we call them genes. Genes therefore are the unit of inheritance. Factors, they are also unit of inheritance. But these factors, no, we call them genes, another name of this factor. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are called alleles. Genes which code for a pair of contrasting traits are known as alleles. That means they are slightly different form of the same gene. Slightly different form, alternatives of the same gene, and they are present in homologous chromosome at same loci. Loci means location. If we use alphabetically symbol for each gene, then the capital letter is used for the trait expressed at F1 generation and the small alphabet for the other trait. For example, in case of the character of height, we use capital T for tallness and small t for darkness, and capital T and small t are allele for each other. They are called as allele because they have some difference. Hence, in plant, the pair of alleles for height would be either capital T, that is pure breed, capital T small t, hybrid breed, small t also a pure breed, right? Mendel also proposed that in the true breeding, tall or dark variety, the allelic pair of genes for height are identical because they are identical, so they are called homozygous, right? Capital T or small t, they are homozygous respectively. Capital T and small t are called the genotype. This one is called genotype because two pairs which type of genes are present. Same types or some alternatives are present. Same type present means we can say homozygous. Alternatives are present, we can say heterozygous. While the descriptive term tall and dwarf, tallness and dwarfness, this one is phenotype, which we can see, right? This is phenotype. What then would be the phenotype of a plant that had a genotype capital T, small t, genotype capital T, small t is there, means we can say it is heterozygous. But what is the question here? What would be the phenotype of a plant? The phenotype of this plant is capital T means what? Tall. The phenotype means tall. Why tall? Due to the dominant character. Right? Okay. From this passage, we will read on the another class. Right? Today we will. Okay, today we stop this class, right? Because our time is over. Thanks.